How does CRISPR work, and why is it important for human health? Today, we're going to talk about CRISPR. CRISPR stands for Clustered Regulatory Interspaced Short Palindromic Repeats. Before we talk about CRISPR, we need to discuss DNA, RNA, and what is called the central dogma of molecular biology. As you have already learned in M3 Science, our genetic code is encoded in DNA by a sequence of nucleotides with the letters C, G, T, and A. C stands for cytosine, G stands for guanine, T stands for thymine, and A stands for adenine. Our DNA has two strands which are called complementary to each other. This means that C on one strand matches with G on the other strand, and vice versa. In addition, A on one strand matches with T on the other strand, and vice versa. Our genome is made up of 3 billion pairs of these four bases. The central dogma states that DNA can encode a copy of itself, except it is an exact opposite copy. This is called mRNA, and the process of making mRNA is called transcription. The mRNA can then leave the nucleus and enter the cytoplasm, where it can produce proteins by encoding for chains of amino acids. This is called translation. Now that we've had a crash course on the central dogma, let's go back to CRISPR. CRISPR was originally found to be a defense mechanism that bacteria use to protect itself against viruses. Viruses attach to the bacteria and can inject their DNA into the bacteria. The DNA merges with the bacteria's original DNA and provides a type of memory against past viral infections. This occurs in two phases. In phase 1, or the acquisition phase, a virus attaches to the bacteria and injects DNA into the host cell. The DNA is incorporated into the CRISPR locus between a series of special repeating sequences. This DNA sequence is then transcribed into a matching transcript called messenger RNA. This messenger RNA joins with two other special pieces. The first is a guide molecule called tracer RNA, and the second is an editing protein called Cas9. These create individual subunits inside the bacterium. In phase 2, when the bacteria is infected with the same type of virus, the Cas9 protein and CRISPR RNA complex recognizes the viral DNA because it is an exact match or complement to the CRISPR RNA. After it is bound, Cas9 causes a breakage of the DNA, effectively getting rid of the viral invader. How does Cas9 perform its function of cleaving DNA in the precise location necessary? This is made possible by short sequences in the DNA called PAMs, also known as protospacer adjacent motifs, which are important in this process. The PAM is a sequence of 2 to 6 base pairs that immediately follows the DNA sequence that is to be targeted by Cas9, as shown here. Here, you can see that the sequence to be targeted is directly adjacent to the PAM. The target location for cleavage of the DNA is identified by an approximate 20 nucleotide sequence that is adjacent to the PAM site. Cas9 will then unwind the DNA strand with the assistance of PAM. This unwound portion of DNA can then be cut, causing a break in both strands of the DNA. Cas9 ensures that the correct sequence is cleaved because the nucleotide target is identified by the PAM. After the DNA strands are cut, Cas9 can glue the ends of the DNA together through a process called non-homologous end joining. The end result is that a segment of DNA has been removed, and the DNA is joined together, making it look like that segment of DNA was never there in the first place. Alternatively, scientists can insert new genes into the DNA using a process called homology-directed repair. In homology-directed repair, a repair template is inserted between the cut segments of DNA and is inserted there. Thus, a segment of the repair template that travels with the CRISPR Cas9 is transferred to one of the two DNA strands. Then, a matching template will be produced and transferred to the other DNA strand across from the repair template to repair the DNA. A researcher can even better target a specific site in the DNA for gene editing. One way of doing this is to use a plasmid, which is typically a small circular piece of double-stranded DNA. It is basically a ring of DNA that can be transferred into a cell. What scientists can do is insert the Cas9 DNA into a plasmid, and then inject the plasmid into a cell in which they want to edit the genome. The DNA is transcribed into mRNA, and is then translated into a protein. And this is the Cas9 protein. Cas9 also needs to be guided to the correct place on the genome to make its edits. Therefore, scientists also inject what is called sgRNA, or a single guide RNA. This sgRNA contains the tracer RNA and the crRNA. As you remember from earlier, the crRNA, or CRISPR RNA, is a 20 nucleotide sequence that is complementary to the target DNA, or the DNA segment that we want to edit out. Also remember that the tracer RNA, like before, is a scaffold that binds the DNA and CRISPR RNA to Cas9. Together, the guide RNA and Cas9 can edit out or excise a defective segment of DNA that might be a cause of a genetic disease. What kinds of diseases might CRISPR be useful to treat? These are genetic diseases caused by a mutation in the genome. These mutations are often passed from parent to offspring through their genes. One such disease is Huntington's disease. It is a progressive brain disorder that causes uncontrolled twitching movements of the body, leading to an inability to walk, swallow, or speak. People who have Huntington's disease start to develop symptoms in their 40s and can die in their 60s. Huntington's disease has an autosomal dominant transmission, which means that their offspring have a 50% chance of getting the gene. So the children have a 1 in 2 chance of getting a single copy of the mutated gene, and that is enough to cause the disease. People with Huntington's disease have a mutation in the Hqt gene. This mutation is caused by a repeating sequence of 3 letters, C, A, and G, which appears several times in a row. Scientists called this a trinucleotide repeat. It means that there are many repetitions of the nucleotides cytosine, adenine, and guanine. In Huntington's, you can have 40 or more CAG repeats in a row. This abnormally long segment in the Hqt gene causes small fragments of proteins to accumulate, bind together, and cluster inside nerve cells or neurons. These clusters of proteins are toxic to nerve cells, disrupting neural function and ultimately leading to the death of the neuron. Scientists use CRISPR gene editing technology in mice that had Huntington's disease. This means that the mice had the same symptoms as humans who have Huntington's disease. Using CRISPR, they were able to edit out the mutated Hqt gene segment. As you can see from this fluorescence image, the green fluorescence correlates with the toxic accumulation of Huntington protein in the mouse brain with Huntington's disease. After using CRISPR, the green fluorescence is much reduced, indicating that the mutated part of the Huntington gene was excised. It is not known, however, if CRISPR can work in patients suffering from Huntington's disease. Experimental trials using CRISPR in humans are currently ongoing, so we should know whether this will be a potential treatment option in people with Huntington's disease in the future. CRISPR has great potential to treat genetic diseases. In fact, the potential of CRISPR was recognized by the Nobel Committee, who awarded the 2020 Nobel Prize in Chemistry to Jennifer Doudna and Emmanuel Charpentier, who were the discoverers of CRISPR. Now you know what CRISPR is, and the next time you hear about CRISPR, it might be in the context of new treatments for genetic diseases for humans.